All right, we're going to talk about alignments and consensus sequences here. So an alignment is when you take two different sequences, maybe they're from two different people or even two different species or maybe even two different genes, but you think they might be related. All of those, if you're what we're doing is we're comparing sequences. It could be amino acids or it could be DNA. I'm going to do DNA here. And so here I have a little sequence, and if, of course, I have an identical sequence, all right, it's basically easy to align these. In other words, the way you see this usually done is there will be like a dash wherever they are identical. So let me put a dash here. It's not a dash, it's actually a vertical line. Let's see if I can get this to there like that. So these two are identical. So I need to put a space here, a couple of spaces. That's a little too many. Let me do this real quick. I think it'll make a difference for being able to see it. Still a little too many. So about 2. 2. There we go. That looks about right. It's hard to get them lined up initially. All right, so maybe, oh, this one could be over 1 maybe. No, that's too much. It looks better there. This one, that looks better. Maybe this one goes, oh, no, that's too much. That looks like a pretty good, so that you can see that the lines, let's see here, show, and then I'll go back on each one. That should work. Okay, so this is kind of showing you wherever there's a line, they are the same. So this is an alignment, and this is a perfect alignment. But let's say we go over here and we have a mutation in one organism and it changes from a T to an A. Now we do not. All right, so we leave that blank. Same here. If this changes to a G, that is no longer an alignment. There we go. So now it would look like this. So this one is close. The sequence is similar. It is not identical, but that's the way you would do this. Now, what happens if there is an insertion? Now, that is a more difficult problem. And let's say we add an A here. This is called an insertion. And so this still aligns. You need to, though, move these over. Right? There. So it looks like this now. Okay, so you'll see something like that, and often you will see maybe they will, certain programs will put a star in there. All right, or maybe they'll put a star down here. Then I have to go back and, of course, do that. And it doesn't line up well with what we're doing here. Let's see, maybe on this side, that'll look a little better. That looks a little better. So you might see something like that. You might also see a dash. So they might put a dash in there. But that's going to make that, there we go. Kind of get it out of alignment when they do that. But you might see something like this in certain software. But the idea is the software, or you, if you do this by hand, you're aligning it. The pieces that go together, and then it will put a gap. So here we have some type of gap. It might be a star. It might be a dash here. You get different things in different places, but essentially that's what you're going to get. All right, so I'm going to do it this way. So I just leave it kind of blank. So we have a blank spot here and a blank spot here. There's obviously something that needs to go here. I don't like that. Let's do it that way. I like that better. That looks better to me. Maybe. Okay, that looks best to me. I'll just leave them blank like this. And we kind of get the idea that, okay, these are aligned, but they're not perfectly aligned. perfect alignment would be all straight lines in there, and you would see that all the sequence is identical. So this is an alignment, and you can do this with multiple sequences. You don't have to just 2 here. You could have 3, 4, 5, and you could have them, you know, the same idea. Down here. So you can see that this one down here is the same here, but not here, but here, but essentially all the rest of them. So here you have, if you were to run this on a software, you'd see something like this. Then you would skip this one. There we go, about like that. A little too much. I think you get the idea. So this one is, which one is better aligned? Well, this one is better aligned with the bottom one than the top one. And the bottom one and the top one are the worst aligned. In other words, if I were to compare this one to the top one, we would get a difference here as well. So there's 1, 2, and then 3. There would be 3 nucleotide changes, 3 mutations if you want, between the bottom one here and the top one. Here there's just 1 mutation, and here there is 2. So, and we also saw that there can be a deletion. All right, so those are usually worse. So it doesn't mean just because this one has 3 changes and this one has 2 between these, which one is the worst? Well, it's this one. A deletion is usually a far worse mutation than what we have when we just have letter changes. So the differences between here may not make a difference at all. Depends. If it's within the gene, it's actually not as bad as if it's in, say, something like a Tata box or maybe even a Terminator. And those are things that we've now talked about or you are doing in the worksheets. So realize you really don't want a mutation in a regulatory sequence. And a regulatory sequence is like the Tata or the Terminator. We'll talk a lot about regulatory sequences. So when you're doing these, though, usually you're doing regular genes. You need to, I should say the promoter, the place where the Tata is, and the Terminators are outside what we used to normally call a gene. But now that we're understanding how it works, we're having to expand the definition of a gene. And so a gene just might not be the protein coding region, but also the regulatory regions around it, which we've just begun to talk about. All right, now this here is an alignment between 3 sequences. Now the other thing we need to know is, okay, we want a consensus sequence. Now a consensus sequence is one where we look at multiple sequences like this, and any place that they're all the same, we say that's good. That's what it has to be. If we see every gene that does a certain thing, say it's insulin, and we see different versions of insulin, and we find a consensus among the many different genes that produce the protein insulin, we put them together and we create what's called a consensus. And I'm going to create a consensus with these sequences. So A. Now, what's the most common letter here? Well, the most common letter here is, we've got 2 C's and a G. Well, it's going to be C. And then here we've got 3 G's. All right, and here we've got 2 T's and a G, so we're going to put a T. Here we've got 3 A's. Here we've got 2 A's and nothing, so we put an A. Here we've got 3 C's. Here we've got 3 G's. And here we have 2 A's. Now sometimes you will see in another notation where if all 3 of them are the same, you kind of make them bigger. So let's do that. Let's make this a little larger than that. There we go. And so here we've got 2 C's. We'll leave this the same. We've got 3 G's, so we're going to make this one larger because it pretty much has to be there. And here we have only 2 T's, so we're going to leave it. Here we have only 2 A's. No, wait. 3A. Oh, nope, that one needs to be bigger. All right, but this one has 2 A's, has 3 C's, so we'll make that one larger. All right, and 3 G's, we'll make that one larger. All right, and only 2 T's. So a lot of times you will also see these color-coded. So maybe if it's, you know, maybe we make the important ones. Let's make it this one red. All right, something like that. Wherever the red ones, we'll make that one red. And I think this actually does make it look better. All right, so what this is telling us here is that there can be some changes within the sequence. The consensus is ACGT-AA-CGT. All right, ACGT-AA-CGT. All right, a little different. But the idea is some are more important than others. And this means that perhaps you can make some changes here in these ones that are not highlighted in red, and maybe it doesn't affect the protein. The ones that are in red, they are more likely to be the conserved ones. Conserved means that it doesn't change over time. In other words, there are mutations from one generation to the next, and you get mutations over millions of years, you get a lot of them. Well, if they tend to not change, that tends to mean they're important. If they're important and they change, often the organism dies or is not fit, all right, and can't survive. So you don't see changes in certain letters very often. So this is telling us that these are the important ones, and these are the ones where the gene has a little bit more flexibility. And that's what we can tell when we do a consensus. So here I showed you a consensus between 3, and we can also tell relatedness. The occurrence of mutations along here happens over the probability of happening at a certain given of time tells us that, well, one of these is probably going to be they diverged, as in the organisms had a common ancestor a certain distance back, and the more changes, the further back the common ancestor is. So, and we can just count it up. So here we only had was it 1 change? The 1 change here. Here we had 2 or 3. Was that it? Or was it 3? I forget what we did here. But I think this one had 2 and this one had 3. So therefore, this organism is thought to be more closely related to this organism because there are fewer changes, and it is more distantly related to this one. And if you were going to count this one compared to this one, they would be even further apart. So this is kind of like a middle ancestor between these two. And that's the way they judge these days. They don't judge by the look of the organism as much as they do the sequence these days. And this is what we use consensus and alignments for. And we'll do a lot of alignments in consensus, and we'll have a big project at the end doing some of this. We'll do some little projects first, some labs, and we'll do a minor project maybe in the middle with this, but then we're going to do a big project at the end using this type of idea. So this is just an introduction to the consensus and alignment, and we'll learn some software tools that'll help us do these things.